Hi, this is Dr. Jabali, and in this video, I'm going to focus on topic 7.2, which is about transcription. I'm going to focus on past IB exam papers related to paper 1, which is multiple choice. So here in the first question, it says here, very soon after fertilization, a parental epigenetic methylation is reversed in DNA. Later, the tissue-specific epigenetic modifications are made to the embryonic DNA. The graph follows the degree of methylation from different sources during the embryonic development. So in the graph here, you can see that there's a degree of methylation and the stages following the fertilization. You have the zygote, the blastocyst, and the embryo. For those who don't remember, when the sperm plus the egg, when they get together, they form a zygote. And the zygote, it basically turns into many cells as it goes through mitosis. So it goes from 2 to become 4, 8, and it just keeps dividing. Eventually, from that, it will become a blastocyst. And as it continues to divide more and becoming a different shape, then it becomes basically an embryo. Now, in this process, we are talking about joining right here the points of mom and dad. So you got mom and dad eggs fertilizing right at this point here, as you see it, and that means their level of methylation is really high. However, right about here, they both start to drop as a blastocyst. This is the father part, right? So you can see that the father has a drop much faster than mom. She does have a drop, but it's a little bit later on after the blastocyst. And then it reach after blastocyst, they both have the drop very low. And that means the embryo has no methylation. But then after that, the development of embryo, it starts to rise the methyl again. So according to the graph, what are the changes in DNA methylation during the embryonic development? So they're talking about the DNA methylation and how it does go through the entire process here. So one says only the parental DNA becomes demethylated. D here means off. So it's not true. You can see that not only dad is going down, mother is going too, so A is off. The maternal DNA becomes demethylated first. If you look very closely here, it's actually the dad that drops first before mom, so it's not B. The methylation patterns of the parent's DNA are erased before fertilization. Now, it's not before. Right here, you can see it's high. It usually is after fertilization that they start to drop, so it's not C. Obviously, only left with D, but we have to read it to make sure the methylation patterns of both parents are erased after fertilization. That is true. If you look from the graph, they both eventually get erased right about here as they reach the point of embryo. So the answer is D. Moving on to the next question, it says, what does the post-transcriptional modification of eukaryotic mRNA includes? When we say post-transcriptional, we are talking about the mRNA editing. And what is that about? That's when you have those exons and introns, and you repeat with the exons and introns in a long strand of mRNA. However, you want to keep the exons and you want to get rid of the introns because introns do not code for anything. And then you're going to join the exons together and the introns are completely gone. After you have joined the exons, then you're going to put a tail as well as a cap. When you do that, then you have finished the entire post-transcriptional modification. So let's look at the options. The first one says introns are removed from the mRNA. True. Exons are joined together to form the mature mRNA. True. The 5 cap and the 3 polytails are added to the mRNA. True. So all of the above, it is exactly happening in the post-transcriptional. So the answer is D. Next question. The variations in the types of antibodies are produced by the mRNA splicing. What is the advantage of this process? What does this mean? So for example, let's say here you have an mRNA and you have exon, let's say 1, and then you have an intron, then you have exon 2, then an intron, then an exon 3, and then an intron, and an exon 4. So you can see here that the exons are the ones that code for protein, while the introns, which are the green here, do not code for anything. Now, when they are talking about splicing, you can make options. For example, what if you want to make of 1, 2, and 3? If you do this, then you obviously would have been cutting, in this case, this one, and all of this. That will give you protein, or let's say antibody A. However, if you make a 1, 2, and 4, then in this case, you probably would have been cutting all of this, but you would be cutting this part and you will leave the 4. So in different locations, you're doing splicings, and this will give you antibody B. Or in the case, you could be also, let's say, you want to do with just 2 and 3. So what would you do? You cut all of this and all of that in the middle and only leave the 2 and 3, and that will give you antibody. So you can see in that one mRNA, you were able to make different types of antibodies. So that's what's the advantage. Now let's look here. It says reduce the size of mRNA required for the translation of antibodies. No, you don't necessarily reduce. That's not the advantage. It has nothing to do with reducing their size. Increase the number of different antibodies that can be synthesized. That makes more sense. As you see, there's different numbers that you can produce. Ensure that one gene codes for one antibody. Now we can see that one mRNA here, which is representing that one gene, can code for so many different antibodies. So it's not C, and speeds up transcription of antibody. It does not speed up because this happens after transcription, so it's definitely not D. So obviously the answer is B. The diagram shows how pre-mRNA is processed into mature mRNA. Which structures are indicated by the letters of W and X? If you look at W here, it's basically the dark area. It still stays on the mRNA. And then you have the white part here, which is highlighted in yellow that I'm doing it, eventually is taken out. So obviously the mRNA keeps the exons and the yellow is the introns. So in the options that we have here, it's not exon poly-A tail exon, intron, as a W, no, it's exon, so it's definitely D. Moving on, gene expression cells is regulated at various stages, during which stage does splicing of RNA occur. Now, this happens after transcription, so it's nothing to do with replication, it's not during transcription, but it says here post. The word post means after. So this is where the mRNA is being matured and is being spliced, so the answer is C. And you can see D is translation, which is after it, so it's not it. Now, how can the environmental factors affect the expression of genes? Now, remember that one gene right here, this one gene can have different types of stimuli. Let's say stimuli A, B, or C. One gene, if it's affected by A, it will produce A protein. If it's affected by B, it will produce B. And if it's affected by C, it will produce C. That's how the environment here can affect a gene to produce different things. So here we have to see which one causes more. By promoting the replication of nucleosomes? No, it doesn't have to do with nucleosomes at all. By inactivating the epinephrine, it has nothing to do with shutting off when you're expressing. By making specific changes to the base sequence of the genes, that would have caused the mutation. So the environment can't cause mutation, but that's rare. That wouldn't be a way of expressing. It would be more of a problem. So it leaves with by causing the pattern of DNA methylation to be changed. See, DNA methylation has everything to do with transcription. So if the environment is changing the way the gene responds, obviously it's going to change the methylation and changing that way the transcription. So the answer is D. Here we have a diagram that shows the arrangements of introns and exons in a segment of pre-mRNA, which that means it hasn't been matured yet. So the pre-mRNA mature containing introns and exons. So you can see here in the drawing we have exons and then we have some of the introns. And if you remember, we try to get rid of the introns. So you only have the attachment of A, B, C, and D together. Now, which is a mature mRNA molecule derived from the pre-mRNA by splicing? So that means what happens after you splice. So here you can see that you have only the exons. So that would have been the right answer. Why I clearly know this? Because here you still have introns, and right here you still have introns, and you still have introns here. So all of these are wrong. You can only have exons. So the answer would have been A. And this is pretty much it, the type of questions that you get for 7.2. So make sure you know about the post-transcription really well, about splicings, as well as expressions of genes. These are main important parts of 7.2.